Hello, everyone, and welcome to Neurocracy. Today, I'm joined by Dr. Ella Striem-Amit, the principal investigator of the Sensory and Motor Plasticity Lab at Georgetown University's Department of Neuroscience. Ella, thank you for joining me today.
Happy to be here.
So, my first question for you is how did you first become interested in studying sensory and motor plasticity?
Honestly, randomly,
Okay.
I was an undergrad in a university studying biology and psychology, and I needed to pick a lab for, an honors program. And I picked a lab that started looking at brain plasticity in people with blindness. And honestly, I never looked back. It's just a rabbit hole of fascinating questions that I just kept at it.
Okay. Could you describe some of your work at the lab and what you guys kind of do there?
Sure. So we study brain organization and brain reorganization
Okay.
in people who were either born with blindness, deafness, or born without hands. And the idea is to go across sensory systems and domains to try and look at the general principles of how our brains develop. And to what degree do they depend on our sensory or motor experience, right? To what
Okay.
degree do the areas of the brain that are usually processing vision dependent on vision to develop their like regular, typical functions?
Okay. So, could you explain what cross-modal plasticity is? So, it's a term I noticed in your lab, and then I have a few questions about that.
Of course. So
Okay.
cross-modal plasticity is like an umbrella term
Okay.
that says we're looking at a brain area that's supposed to be processing vision, for example. So it's supposed to process the visual modality. If it's processing instead the auditory modality or tactile modality, then it's performing some sort of a cross-modal plasticity. It's changing its function from one sense to another sense.
Okay. So, in your studies of blind and deaf individuals, has there been anything revealed about cross-modal plasticity?
So yes, so first of all, in my field, before my lab started, people have already been doing really, really beautiful work showing that visual cortex in people who are born with blindness can, I'm kind of hesitant to use the term reorganize, but can
Okay.
kind of process information from other sensory modalities. But what it seems to do is process the same type of information. So an area that's supposed to be dedicated to reading in all of us would simply be dedicated to reading in Braille. An area that's supposed to be processing, I don't know, faces will still be processing identifying, understanding people or identifying people or their emotions through other senses. And the same goes for people who are born with deafness. So their entire language system, part of which is supposed to be in the auditory cortex, is still processing language just through signing.
Okay. So, could you explain how you separate innate brain organization from experience-driven changes?
That is a big question.
Okay. Yeah.
So honestly, I kind of think of it as okay, so the original question is supposed to be like nature versus nurture, right?
Okay.
Like what is the cortex dependent on experience to develop, and what is it there that's like innate, that's at the baseline when you're born, even before you have sensory or motor experience, right?
Right.
And so the question is, what is everyone born at with? How is the brain organized at birth? In a way that, regardless of whether you have experience with a specific sense, it still has that same type of organization. So the idea is, for example, that the connectivity of the brain, a lot of it develops before birth. So it would stay pretty much the same if even if you're born without sight and you don't gain sight, but you're a lot of the brain wiring is already there and stays that way. In contrast, experience dependent plasticity is the development of functions that really rely on you having that experience as a child. Does that make sense?
Okay. Yeah. So here's the thing about that, actually. This is kind of a follow-up. So one of the reasons I was interested in your lab's work is I am actually a speed cuber. So I just wanted to ask, like, would you be
You're
able to
what?
see?
Sorry?
I'm a speed cuber, so I use like Rubik's cubes,
Ah,
and I can solve one in I've solved one in 4 seconds unofficially, but I average about 7 seconds. So I've spent years like just turning these cubes for hours and hours, and I wanted to
oh
ask,
yeah.
Do you think that there would be a change in my brain compared to someone who's never touched the Rubik's Cube?
I am sure there would be.
Okay.
I'm assuming that your representation of the Rubik's Cube and the spatial relationships
Yeah.
and like the way that moving it orders things around, your spatial, visual spatial memory
Okay.
would be really, really strong in general and specifically with the type of computations that you're used to performing with this.
Okay.
I'm assuming that if I had like a test that measures it kind of regardless of the Rubik's Cube,
Right.
we'd be able to still pull that apart.
Okay. That's interesting. I like asking about that because that was one of the things I was wondering in the past few years as I've been kind of studying my own like neuroscience and thinking about cubing. And I was like, I wonder like what kind of like skills I'm developing in my own brain. So my next question is kind of about your findings. And do you know like how your findings could potentially guide the design of better prosthetics or rehab protocols?
So far, what I've talked about is mostly about stuff that is in my field and not specifically in my lab. So in my lab,
Okay.
what we do is study brain organization for actions in people born without hands. So we're looking at the motor system and we're looking at, let's put it this way. If I'm grabbing my coffee cup,
Yeah.
what I need to think about in order to do that is less to fine tune like how big the aperture of my hands is or my fingers, and more like have a general idea of I want to bring this cup, grasp this cup and bring it here, right?
Yeah.
And that is a way of representing actions in the brain that is kind of slightly understudied. Okay.
Okay.
A lot of people study the fine motor kinematics and how the brain controls those. A lot of people study the high level behavioral goals. And what we're trying to study with the aid of people born without hands is whether we have this like mid-level representation of action types that doesn't really care about the fine motor specifics. In other words, I can grab my coffee cup with my hand, I can grab it with my mouth. I'm not going to demo this because I am definitely,
Yeah.
definitely going to spill it all over me on video.
Right.
But people born without hands can do that with their feet because that's how they, manage everyday life.
Yeah.
So by looking into how those actions are represented in the brain, we're hoping we can figure out how that level of representation exists in the brain, and then to be able to tap onto that to design prosthetics and brain-machine interfaces that specifically are tailored to reading that information, and
Okay.
use that to augment the current type of prosthetics.
Okay.
So that's one thing. The other thing is that we study differences, individual differences between people, right? I've been talking generally about blindness and about deafness, but like every individual is unique, right?
Right.
One person with blindness may be really adept at reading Braille. The other person may be more into audiobooks and doesn't use the tactile modality or touch that much. Another person may be really, really good at memory and memorizing paths, and that's how like they get along in daily life. And we're trying to find like the neural correlates of
Yeah.
that to be able to identify who's who, and to be able to then say, okay, if this is how your brain is wired, maybe you'd benefit from these tools more than those tools.
Yeah.
Maybe that would make you a candidate for this type of rehabilitation versus another.
Okay. Is there any sort of like genetic correlation by any chance, like in these kind of like differences in abilities?
So we haven't gone to that level of analysis yet. We're
Okay.
looking
Okay.
at brain connectivity patterns.
Yeah.
People have been looking at genetic differences. Um, there are some correlates, but like again, another field of research that there's
Right.
lots of room for.
Yeah. So I want to actually bring up a myth that kind of came to my head as you were explaining that. So I've heard somewhere or just many places I guess that whenever like one of your senses stops working, like let's say you're deaf, like your other ones get enhanced. Could you like say if that's true or not and just bust the myth either way?
So I know it comes across as a common myth, but actually on average it's true.
Oh, okay. Okay.
Yeah.
Wow. Interesting.
On,
Could you explain
yeah,
like some of that, I guess?
of course.
Okay.
So I mean, two things, right?
Okay.
First of all, people who are, for example, born with blindness have more practice of using, say, audition for understanding space, for understanding things around them, right?
Yeah.
So they need to extract, and they train to extract, and they then extract more information out of sounds. So it's not like it's it's not magic, right?
Right. I
They
guess, yeah.
work at it, right? They use it more, they need it more. And so they train at it more than we need to.
Okay.
Um, and the same goes to some degree to people with deafness. There are specific subtle differences and advantages that they have in attention to space.
Interesting.
Uh, so it's kind of true, but again, it's on average, right? And because
Right.
every individual is different, you can't kind of assume that
Yeah.
everyone is.
That's kind of like the mean out of like a lot of like data, I guess. So my next question is hopefully more interesting, but I want to ask kind of like the technology that you guys use in your lab specifically, or if you want to talk about the field in general, whichever you think is good.
So we use fMRI, which
Okay,
is kind of
that's what
a
I figured.
very
Yeah.
established technique, right?
Yeah.
Do you want me to go into explaining this, or
Yeah, sure.
you're
I think that would be interesting. Yeah. And then you can explain the fMRI a little bit, and then you can talk about like I guess the field in general if you want.
Sure.
Okay.
So fMRI is just a very simple, very established, very safe technique to record brain activity in people as they are lying in the scanner and mostly chilling. My son is going to be doing an experiment in 2.5 hours also.
Oh,
It's very safe, very
okay.
reliable, non-invasive. So you lie in the
Right.
scanner, you're either presented with stuff that you see, you hear, or you need to do. And and then we model the brain activity because we know the fMRI allows us to track the balance between oxygenated and non-oxygenated hemoglobin.
Yes.
Um,
Okay.
and so we know that right after you've maybe seen something, the visual areas in your brain will act. They'll use more oxygen. You'll get a need for more oxygen in the brain, and then you'll have an influx of more blood with more oxygenated hemoglobin. And we can track that increase in blood flow and oxygenation level to say, oh, that area is the area that was just now active. That means it responded to the stimulation, to the visual stuff that we showed it right before.
Yeah. So what are some examples of like tasks that could be done by someone in an fMRI and used to like study?
So we can do a lot of, well, anything that doesn't require you to move too much.
Okay.
Because we're taking pictures of your brain,
Right.
right? So we need your brain not to move.
Okay.
And we need most of you to be kind of stable, like not do a lot of things other than the things that we're asking you to do.
Okay.
So, for example, we do experiments in which we present sounds like words or sounds of animals and scenes and stuff like that, or even simple tones to map the auditory cortex. We have like those buzzing things that stimulate the fingers, so we can
Okay.
present touch as well. We can present I said sounds, so we can present images or videos or
Okay.
whatever we want. And we have people moving in the scanner and doing simple hand and foot actions
Okay.
in the scanner.
So you might see where this is going, but I want to ask, is it feasible for me to like go into an fMRI and like turn a Rubik's Cube and scientists to see like where in my brain, like where like which parts are being active?
Yes, it is completely feasible.
Okay.
The challenge would be to try and isolate what it means, right? Because as
Okay.
you're doing this, you're both seeing things and moving things. And like there's so many processes going on that if I just look at your activation as you're doing this versus not doing anything at all, most of your brain would be active, right?
Right, right.
So a lot of like the challenge in experimental design is
You're just trying to
designing
control
techniques.
it as much as you can. Yeah. So
Exactly.
like perhaps like this is just completely hypothetical, but like I
Yeah,
could potentially just hold it and stare at it maybe. And then like in another one, like I could be turning it and then that could maybe like kind of level out some of the variables to create like a more controlled like analysis of what's
exactly.
going on. Interesting, interesting. It's something
Exactly.
I've
And
always wondered about and I didn't expect to kind of talk about this with you today. So that's good. Is there any other technology that is interesting and used in your field?
so for what we do, we're mostly doing fMRI. We're doing some behavioral experiments as well,
Okay.
right? Like we want
Yeah.
to correlate what we're seeing in the brain to what their people's more everyday behavior like, right? So other
Yeah.
measures as well. We're tracking hand and feet movements to correlate that to the motor activity in the brain as well.
Yeah.
So you're doing some kinematic tracking. That's most of what we do. Of course, in neuroscience, you can also have electrophysiological measures like EEG on the entire head,
Okay.
or even implanted electrodes in specific clinical cases. There's a lot of like neuroscience techniques that one can employ,
Okay.
uh, even in human research.
Okay. So, in your years of research, and it doesn't necessarily have to be in this field, but what has been like your single most like favorite finding or experiment or kind of problem, maybe even?
Uh, yeah, no, I can't answer that one, right? Like it's like picking which one of my kids
Okay.
do I like best. Uh,
Yeah. Yeah.
I have two research lines right now.
Okay.
One about individual differences that I'm super, super curious about. And
Interesting.
the other one is about action representations. And I can't like say which one is more fun to work with right now.
Okay. Could you pick one maybe and just talk about it a little bit? I think it would be interesting to hear about. I can hear like your passion for the first one especially. So,
The individual differences.
yeah, I'm curious. I kind of want to just hear a little more about it.
Sure. I feel like I know less about what we're gonna find there, so it's a little more exciting. It's more
Okay,
preliminary.
that's good. Yeah.
So so the situation is kind of the following, right? Like there's been research in the domain of looking at brain plasticity and brain organization in blindness
Yeah.
for decades now. People have done beautiful, beautiful work, and they've shown that the visual cortex in people who were born with blindness can really respond to a lot of things, right? So like you were saying, okay, if I were to cube in the scanner, which parts of my brain would be active? And for you, definitely the visual cortex would be active because you're looking at it. But in
Right.
people born blind, right, their visual cortex is active for almost any task that people tested in any
Uh,
sense,
okay.
which is super confusing,
Right.
right?
Yeah.
So it's active when they hear things, when they touch things, when they read, when they hear language, when they do math problems, when they try to echolocate, even when they smell things at
Okay.
the group level. And it feels to me like it's more confusing than insightful to say that, right?
Okay.
Because like, yes, it's active, but why, right? What is it doing there? So now we're testing this hypothesis that it's the reason it's active for so many things at the group level is because different people may be using it differently. And
Okay.
that's the reason we're looking into individual differences. We're going to try and see whether, if you're born blind, your brain, the visual parts of your brain that are left without the natural, typical quote unquote input can actually reorganize for many different things. And it's probably a combination of your genetics
Yeah.
and like what you're training at, like what is it that you're trying to compensate with that kind of drive the organization of these areas.
Okay. So in people born blind, would you say there's that important like plastic period, like those periods during like childhood?
Critical period.
Is it like
Yeah.
yeah, I forgot the name. The critical periods, like is there significant development in kind of like understanding like even without like the vision like sense, like still like understanding for example how to like read like using like Braille and I guess could you just explain I guess the significance of the critical periods in their
Sure.
development? Yeah.
So generally critical periods or sensitive periods, because they're not like as clear cut as
Right.
we initially thought, are just periods in which the brain is like super sensitive to receiving the right kind of input for it to develop the right kind of architecture for it.
Yeah.
In other words, if you have amblyopia, if you have a lazy eye,
Right.
and one of your eyes is needs glasses and you're not getting those glasses in time, your brain will not interpret information coming from that eye because it can't integrate it. It's the least efficient eye.
Yeah.
And over time, if you continue not treating it, you may get to a point in which the brain kind of like gives up. It kind of stabilizes at a state in which it's not really using that input. And that closure of the critical periods means that the brain is no longer trying to optimize for that input.
Right.
What it means with relation to blindness, what people thought it meant with relation to blindness, is that if you don't receive vision at all in the first years of life, you will not be able to see anything, even if your eyes themselves are completely fixed later.
Uh,
And people have been showing more over the years, it started with Pavan Sinha at MIT and continued with other groups around the world, that even if you regain or gain sight later in life, you can actually learn some things. You can still learn to see to some degree. So we know that the critical periods aren't like super, super, super strict. We
right.
know there's a little bit of wiggle room. We know that not receiving the input to some degree may keep things more flexible than they otherwise would. But there's a lot of open questions about like how far can you push that metaphor, or whether if you don't have sight, you can't learn to see X and after a certain period. So that's the concept of critical periods.
Okay.
Now, the implementation of it is very confusing because each part of your brain or each system has its own critical developmental period. It kind of depends on when they're supposed
Uh,
to develop to begin with.
so each one has a different, slightly different, like each part in your brain.
Yeah.
Oh, wow. I actually didn't know that. That's interesting.
So for so the simpler part of the brain that process simpler features are things that really should be like think about animals as they are born. A lot of them are born with eyes open because they already need to process information, right?
Oh, okay.
So in a lot of ways, low-level perception needs to stabilize really fast.
Right.
But there are things that develop really late in all of us, right? Like think about emotional regulation,
Yes,
right?
that's
We
exactly
develop,
what I was thinking about. Yeah, that's the
right?
first thing in mind. Yeah.
We're developing the frontal lobe, we're developing
Right.
emotional regulation, we're
Right.
developing all those things until, and rationality, we're developing those until
Yeah.
like we're 25.
Yeah.
So these things have critical periods too, but they close way later.
Yeah. All right. So, on that note, thank you for joining me today and thank you for sharing your insights with the podcast. I think we had a great conversation and it was great to finally ask someone about the Rubik's Cubes in my brain. And it was a pleasure having you on.
It was pleasure having talking to you as well. And now I'm going to spend some time thinking about controls for Rubik's Cube in the scan.
Oh, yeah. All right. Everyone, subscribe and subscribe to my Rubik's Cube channel as well.